Hey guys, welcome back. So today, first and foremost, I'd like to apologize for the subpar audio quality. A, I'm in the classroom with a lot of echo, and B, I forgot my microphone thingy before. So yeah, bad audio, sorry. But today, I'm going to talk about linked genes. And these are genes that are on the same chromosome. Right? And so as humans, we have 23 chromosomes, right? And every chromosome has a whole bunch of genes on it. And so when we usually talk about inheritance, we think of genes as being kind of like independent things that can move about on their own. But a lot of genes are actually physically tied together by the fact that they're on the same chromosome. Right? And so the inheritance is no longer properly predicted through probability. Right? What that means is that our Punnett squares don't work for linked genes. Right? Because when meiosis happens and they are separated and spread out through the process of meiosis, right, the genes that are on the same chromosome are always going to move together. Right? So they're going to be linked to one another. And so if you have two traits that are linked, let's say we're dealing with eye color and height, right? And we have the alleles for eye color and height on the same chromosomes. Right? This combination of dominant eye color, recessive height is always going to get separated together through the arbitrary assortment process of metaphase 1. Right? And so the big D and the little e will always move together into the gametes of this individual. Right? The little d, the big E will always move together into the gametes of this individual. So they're not independently assorted, is what that means. They move as pairs. The only way to separate linked genes in an individual is through a crossover event. So if crossover happens to occur between the two linked genes, in this space in between, then we get a recombinant chromosome with the different combination, right? So the big D could be with the big E in this instance. If crossover just so happened to happen between these two genes. Okay? And now, the closer the two genes are on the chromosome, the less likely crossover is to occur between them. Right? Crossover occurs at different spots throughout the chromosome. And so if there's only a little tiny itty bitty space between the two genes, the probability of crossover happening in that little space is low. Right? So those genes are going to stay together more often than not. If the two genes are at polar opposite ends of the chromosome, anytime crossover happens to that chromosome, they'll be separated. Right? And so the further apart they are, the more recombinant versions you'll see in the offspring. So when we're talking about linked genes, we're talking about multiple traits together. And in order to really understand how to predict these linked genes, or rather to assess whether two genes are linked, we have to look at how to do multiple trait Punnett squares. So you should learn about this freshman year, but you probably want a refresher. Right? And so this is when we are looking at two traits together. Okay? It's not like we're looking at eye color and height. Okay? We're looking at eye color and height. Okay? One thing with two traits together. So possible phenotypes for these two traits together would be tall with blue eyes, or tall with red eyes, short with blue eyes, or short with red eyes. Okay? They're together. So this is one phenotype. This is another phenotype. This is another phenotype. This is another phenotype. Okay? And our genotypes are also together. Okay? And so I have my A's and my B's, my A's and my B's. This is one genotype, even though it involves two different genes. Okay? And there's a bunch of different options for this. I didn't feel like writing them all out, so I just gave you a little sample here. Okay? So this would be homozygous dominant for A and B. This would be heterozygous for A, homozygous dominant for B. So if we're making a cross with this two trait setup, okay, and I got one of my mates is heterozygous for both traits, and one of my mates is homozygous recessive for both traits. When these organisms do meiosis, okay, one variation of A and one variation of B is going to end up in each gamete. And my Punnett square has to represent all possible gamete combinations. And so you're going to FOIL these, right? That's one way to do it. So you go big A, big B, big A, little b, little a, big B, little a, little b. Okay? And so those are going to be your gametes over here. You're going to do the same thing for this. Now, homozygous recessive means they're all going to be this, right? And for the sake of this first example, I've written them all out. Moving forward, if you have copies of these, right, like they're all the same, you don't have to do this whole part. You can just do the one and slide it down, because it's all going to be just repetitive. Okay. So if this gamete meets this gamete through the process of fertilization, this would be my offspring. So it's big A, little a, little a, big B, little b, little b. If this gamete meets this gamete, this will be my offspring. This gamete meets this gamete, this will be my offspring. If this gamete meets this gamete, this will be my offspring. And now these would be repeated over and over again, right? So this is the same as what I'd be having here. It would be the same here, the same here. Okay? And so ultimately, statistically, that would be 4 of 16, okay? Which is the same as 1 of 4, which is why you don't have to do any of these. Okay? So my genotypes are here. Here are my genotype rates. So 1 out of 4 was this genotype, 1 out of 4 was this genotype, 1 out of 4 was this genotype, and 1 out of 4 was this genotype. My corresponding phenotypes, right? This genotype would be this phenotype. It's going to be blue and tall. Blue, tall. Okay. Blue, short. Short, wait, what? Red, tall. Short, red. Okay. Red, short. So my phenotypes are also 1 out of 4. Since all of my phenotypes are 1 out of 4 probability, okay, theoretically, if these two organisms were to make 100 babies, okay, about 25 of those babies should be each of the phenotypes. Right? So I should have approximately 25 blue tall babies. I should have approximately 25 blue short babies, approximately 25 red tall babies, and approximately 25 red short babies. Okay. Now, because it is random, I might have 20 blue tall babies and 30 blue short babies. That's not statistically different than 25 and 25. Okay. It's not going to be exactly 25 and 25. The larger the sample size gets, the closer it will be to being the precise estimations. But it's close enough, right? However, if these two organisms were to mate, okay, and the babies came out 50% blue and tall, 0% blue and short, 0% red and tall, and 50% red and short, that is statistically different than the expected outcome. And one will look at that and say, well, why did that happen? And the answer is gene linkage. Right? Because the two alleles were tied together during meiosis, when they're separated into gametes, they move together. Right? And so those linked genes, those linked alleles, are going to be this version and this version. So in this case, my linked alleles would be these things linked, right? So my little a is going to be linked to my little b's. And in this case, my big A would be linked to my big B, and my little a would be linked to my little b, right? Okay, so they're linked. And that's how it's going to happen. It's going to be 50%, 50%, and then there'd be none, right? And that's an imaginary scenario where there's no crossover happening. Right? So if there's no crossover, this is what linked genes would look like. 50% one version, 0%, 0%, 50% the other version. The higher the percent of the two recombinant variations, right, the further apart the linked genes are on the same chromosome. Because the further apart they are, the more likely crossover is to separate them and to make those recombinant variations. So if I have this set instead, I have it be 45%, 5% recombinant, 5% recombinant, 45%, versus 40%, 10% recombinant, 10% recombinant, 40%. The one with the larger amount of recombinants are going to have the genes further apart. And so we can use this fact to map out the relative distance between genes on a chromosome by looking at how many recombinant babies are made during an experiment, right? And so we take two of the organisms, we make them make babies, and then we count how many babies they got. And then we're like, well, these ones are 10% recombinant, so those genes are 10% apart. Right? This one's 25% recombinant, so those genes are 25% apart. And we can map the distance of the genes on a chromosome by doing this. It's an estimate, right? But it works. Right? And so that's it. That's gene linkage. Two genes are on the same chromosome, so they are not separated through independent assortment. So we do not get a fair prediction using Punnett squares. Punnett squares don't work for linked genes. That's it. Okay?